Welcome to Brainstorm, where we give you a glimpse into the world of science for this Monday, May 7th, 2012. Our top story comes from the world of biotechnology, where researchers from China have developed a new and improved method for producing genetically engineered animals. Before we get into this method, let's explain the old one, which starts with the creation of chimeras. First, genetically modified embryonic stem cells are injected into the blastocyst of the animal, usually mice. The blastocyst is a very early stage of embryonic development, so that one modified cell multiplies and is randomly incorporated into the animal's body as it develops. When trying to study what a particular genetic modification does, they had to breed these chimeras and just hope some pass on the modification, resulting in offspring with only modified cells. But with the uncertainty inherent in this process, a better one is needed, so the researchers produced haploid embryonic stem cells. These special embryonic stem cells are made by removing the nucleus of an immature egg cell, then injecting it with a sperm. And these are able to be directly genetically engineered and gain many traits of sperm so they can actually be used to fertilize eggs. The researchers successfully produced mice from these haploid stem cells, and it's a much more reliable method of genetic modification. This should assist in medical and other research, also potentially allowing for the engineering of animals not possible in the past. Next, we turn to the world of medicine. A study by scientists in Italy have found a way to improve a new, minimally invasive cancer treatment. Called irreversible electroporation, it's essentially zapping holes in cancer cells and has shown effectiveness against many soft tissue cancers. It's starting to be used with difficult-to-treat tumors. It works because a large enough hole in the cell's membrane automatically triggers its suicide mechanism. But it's not 100% effective, so the team from Italy began testing to see if nanotubes could boost this new treatment. After their initial trials, a former staff scientist at NASA's Langley Research Center read about it and gave the team a sample of extremely high-quality nanotubes. These were highly crystalline with few defects, ideal for this kind of medical research. Varying concentrations of the nanotubes were put in a solution and placed on human cancer cells in a petri dish. A certain concentration even killed 25% of the cancer cells on their own. But the real test was using the tubes with electroporation treatment. What resulted was over doubling the effectiveness. Without the tubes, the treatment killed 40% of cancer cells in the petri dish, whereas with the nanotubes, the treatment killed 88% of the cells, and animal testing is on its way. We end with an update from the world of biology. A newly discovered microorganism from Norway may belong to its own kingdom. For some review, a kingdom is one of the most general categorizations there is in standard taxonomy. Now, this organism is categorized as a protozoa, but that just means a eukaryotic cell that moves around. Most protozoa are difficult to culture, meaning few genomes have been sequenced. However, this organism from a lake in Norway was able to get genetically analyzed. What the scientists studying it found was that it didn't genetically fit into any kingdom of life. Instead, it likely belongs to a separate but very old branch on the tree of life, probably with other protozoa that haven't been genetically analyzed yet. It's estimated that this organism hasn't changed much in about 1 billion years, making it the oldest eukaryotic organism discovered. With traits of both protists and amoebas, this organism certainly has interesting implications for evolution. Well, hope you enjoyed this episode. If you did, please consider subscribing and be sure to check the links in the video description.